I'm going to be talking about how to use anatomy in surgery. Gwen was talking a lot about technology. We have technology every day coming in, but this technology is not available in the whole world. Where I live, South America, I live in a 20 million people city, and maybe we have 5, 6, 10 neuronavigation systems. And I've been traveling all around, and I see that many countries don't have all this technology. Fortunately, I work in a very good hospital that has all this technology, most of it, but sometimes it doesn't work. It's very common to have the neuronavigation system, and I said, there's something wrong here. Or I'm seeing the ultrasound, and I don't know exactly where I am. And then we are in the middle of the tumor, and we put the neuronavigation and said, well, we cannot trust this anymore. And then it's common to come here, and you guys are going to be doing a craniotomy. You see the tumor, you're using the stealth, the neuronavigation, to know where to put a craniotomy. What about if it doesn't work? What about if you have to do some surgery somewhere? The right thing to do is to know where the lesion is. I always say that a neurosurgeon might not know what the lesion is. Which lesion is that? But we have to know where it is. That's where we work. We have to know where it is. So we're going to be talking about this, anatomy, and how to use anatomy to help our surgery, and in many cases, not doing only with anatomy oriented. Okay. Now, I always, in these courses, I divide in two parts. The first part, I talk about what I call the architecture of the brain, talking about anatomy itself. And the second part, tomorrow, we're going to be showing actual cases, how we can use this anatomy in applying to surgery, okay? And the last thing is, sorry for my English. Sometimes the words doesn't come very well. They don't come very well. But fortunately, the images will talk by themselves, okay? All right. I'll start with some 2D slides. You don't have to put your glasses, because I wanna talk about some theoretical issues that is going to help you very much to understand why the brain has this shape, why the structures have the shapes, and why they have certain very constant relationships. So let's understand some developmental issues, okay, of the brain to start with. Fortunately, we don't have a lot of pressure to finish the lecture, so we just go and finish and then continue, okay? All right. As I said, neuroanatomy can be and should be studied by very different points of view. But for all of them, you need to know morphology. For all of them, you need to do morphology. You have to know morphology to understand every other types of neuroanatomy points of view, of course. And that's what we're going to be talking about. Okay, when you're going to operate this lesion, okay, just like you have to know that Italy looks like a boot, that Rome is in the occidental side of the boot, okay? So you have to know that the lateral wall of the anterior horn is the head of the caudate. Okay, and then if you're going to do surgery in this, you're going to have to go as much as you can through natural spaces, okay? So you have to know that to go through natural space, you go into hemispheric fissure, you're going to find the corpus callosum, you're going to open, you're going to head into another empty space. You have to operate as much as you can in natural spaces, of course, as any type of surgery. If you get in the ventricle and you see the choroid plexus, you have to know that you are not in the anterior horn, because the anterior horn is anterior to the foramen Monroe. Anterior horn doesn't have choroid plexus. So if you see choroid plexus, you have to know you are in the body of the ventricle. So you just put your microscope more anteriorly, and you can see foramen Monroe. And of course, you have to know that the lateral wall is the head of the caudate. That's where your lesion is, and you can remove it. Of course, you don't need a neuronavigation system to do the surgery. Of course, you don't need. Okay. So we're going to be dealing mostly in surgery with space and shape. When you see an MRI, you go from the CSF spaces to the lesion. You recognize the sulci, you recognize the shape of the ventricles. That's how you start looking at MRI, okay? That's why we talk about brain architecture, and that's what we're going to be talking about. We have to know the relationships of the structures with each other, particularly with the CSF natural spaces, particularly to do microneurosurgery, and of course with the skull, in order to know where the lesion is, where to put our craniotomy. So the shape is something very important for us to understand. You recognize the sulci, you recognize the shape of the ventricles. Another very important thing to have in mind is that, unfortunately for us, the brain, the human brain has a lot of vestigial structures. These structures were very important at some point of evolution. But now they are not functional more important anymore. But they are still there. And they can be landmarks for us. Okay? And the anterior commissure is the better of these examples. You see, it was the main commissure in very old reptiles. Current monkeys have only 5% of all commissural fibers. Humans have just 1 to 2% of all commissural fibers. The density of fibers of the anterior commissure is much more dense than the corpus callosum. And look at this. The anterior commissure has 3 times more fibers than the optic nerve, which has 1.2 million fibers, and which is the main cranial nerve we have. We are visual animals, okay? And just for you have 1.2 million fibers in the cranial, in the optic nerve, and the optic nerves have 40% of all the fibers that there are in the cranial nerves. So optic nerve is very important, as you know. And anterior commissure has 3 times more fibers. So, and it's not important anymore functionally. It might be for something we don't know exactly what it is, maybe functionally. But if you go there and cut it, if a tumor is there, that's not a big deal. So we're going to be dealing with a lot of vestigial structures. And that's why it makes neuroanatomy very difficult, because we have all these very small, some systems of fibers that we don't know if they are functional anymore or not. So let's start understanding that the first vertebrate animals, the very primitive fishes, they have a very, very horizontal, linear CNS. Okay, now let's call this the cord. Let's call this the hypothalamus that did control their internal functions. And let's call this the striatum that controlled his very poor movements, primitive fishes. Now, when these animals come to Earth, a lot of things happen. Imagine to adapt to a new temperature, to deal with the body without being in the water. Lots of things happen. You can imagine this. But other than that, these animals needed to perceive this new world. Okay? They have to perceive. And they developed for this the olfactory system. They developed olfaction. So they developed structures that they could smell. Now, they had to store the memories of these informations that they were taking from this new universe. Okay? So they developed the hippocampus that has 3 layers, and they could store the memories of the smells. If it was something good, something bad, if it was something poison, if it was a sexual partner, whatever. They had to recognize this. And when they recognized through this storage, they had to have their behavior directed to something. Let's go for it, or let's get out of there. Okay? And then they developed the amygdala that was sort of telling them what to do in relation to this new information. So these structures, these 3 structures, the olfactory structures, the amygdala, and the hippocampus, they arise together throughout evolution, pretty much together. And they arise around the top of this linear CNS. That's why they are called limbic, because limbic in Latin means around, means ring. So that's why it's called a limbic, just because of this, okay? Now, when the mammals came, mammals have had a lot more of information to deal with. So I always say, if you live in a very small town, you can have telephone in each house. But if you live in a big house, you need a telephonic center to receive the calls and then to send to each house. That's what the thalamus is. So the thalamus that receives all this new information to send to the cortex, to discriminate this information, they came along together. The cortico-thalamic system, it arose together when the mammals came, okay? So that's how these structures came. Now, regarding shape, you see that it started with the linear shape, and then it started to bend around the center. So this bending process was very important because it was to save, not to have a very big volume. And then when the sulci developed, it was again an unfolding process towards the center. And we're going to be seeing that the center is the thalamus, okay? And that's how the surface of the cortex did increase threefold. So that's what nature did in order to save space, to make it grow. So this is very important for us to understand why the structures have particularly the deep structures, the subcortical structures, have the C-format structure because of this. And it's very interesting that when we go through embryology repeats at least the most important steps of all evolution. So this bending process was very important. So we have the thalamus pretty much in the middle. The thalamus is not only our functional center of the brain, but it's also our morphological center. We start from there. In the lecture, we're going to be starting from there. You remember the story of these three vesicles of the neural tube, and you remember that the structures did arise from the surface of the tube, while the center did develop into the ventricles, the aqueduct, and so forth. You know about this. But all this happened with the tube bending around itself, okay? So the prosencephalic vesicle gave rise to the lateral ventricle. The inferior aspect of the telencephalic cavity gave rise to the third ventricle, and then you have the aqueduct, you have the ventricles. That's why it has the shape. It's a C shape, okay? The posterior horn is very variable in everybody, very variable. Some people have a very small posterior horn. Now, if you understand what happened to the hippocampus, you're going to understand why the inner structures have the shape, just by understanding this. As I said, the hippocampus, originally, it was in the top of that linear CNS. During this evolution, while the C-shaped format of the vesicle, the prosencephalic vesicle and so forth, was adapting the C-shaped format, what did the hippocampus? The hippocampus did come inferiorly here. It just lied here until it came to what nowadays it's inside the inferior horn or temporal horn. But what happened? When this thing came down, it left the tail. The tail of the hippocampus is the fornix. Okay, so we're going to be seeing that the fornix is the continuation of the amon horn, which we call the hippocampus, okay? So, and this happened around the thalamus. That's why the fornix does exactly this format, just because of this. It's an evolutionary issue that happened here. Now, this happened when the mammals were developing. The thalamic and the cortical system were developing. So the corpus callosum was also being developed, because as you know, corpus callosum put together the two hemispheres. So this happened at the same time that the corpus callosum was being developed. And there was a vestigial structure of the old hippocampus that was left here. And this vestigial structure is the indusium griseum, okay, that you never understood. I never understood until I learned this, okay? So the indusium griseum is exactly here is the vestigial, ancient, very ancient hippocampus. Now, of course, then, it has a continuation of the hippocampus along the so-called gyri fasciolaris, another very small gyri. So that's why this vestigial structure is here, and the fornix is here, and this part is not functionally important anymore. You know that this part is very important for memory, short memory issues, okay? But that's why this has the shape. We are here, number one, to understand the shape. Winthrop said a lot about functional. You're going to be talking a lot about functional. But as everybody says, if you don't know geography, you will not understand history. Okay, so you have to know geography in order to understand the function. So, and to understand the geography, you have to have in mind the shape of the structure. So that's what happens. If you understand this, you understand why the structures have the shape and where they are. So what we call the hippocampal formation, which is pretty much all these structures together, they had the C-shaped format, of course, around the thalamus. And what happened to the amygdala? The amygdala just stayed anterior to the head of hippocampus and covers the head of hippocampus. So, of course, the amygdala will be the anterior wall of our inferior horn. Very interesting what happened to the striatum. If you understand, you're going to understand the projection fibers, the internal capsule. What happened to the striatum? Again, it was piece of mass of tissue in the top of the very old CNS. And it bent like everything, okay, and it did acquire the C-shaped format. but it happened at the same time that the thalamus and the cortex were developing. So projection fibers were being developed to go up and down, take the information from the thalamus to the cortex, and from the cortex to the brainstem and to the cord. Okay, so what happens with these fibers? You see, we had a single striatum to start with, and when the projection fibers started to develop, they did divide. They split the corpus striatum. They split the corpus striatum. That's why part is medial and part is lateral. But how did it happen? As you know, the fibers have a fan shape because they are going towards the brainstem. They are going to constitute the brain peduncle, the midbrain peduncle. So the dorsal part, we call dorsal everything that is behind, is posterior in anatomy. The dorsal part was split, it was divided. But the anterior part, what we call in anatomy the ventral, a ventral is everything that is facing down, it's anterior. The ventral part was not split. So the dorsal striatum, okay, resulted in the caudate nucleus medially, in the putamen laterally. Now, the ventral striatum was not divided. And what is there in the ventral striatum? Ventral striatum is pretty much synonym of nucleus accumbens nucleus. Pretty much almost the same thing, okay? So here's the nucleus accumbens. So that's why this thing has this very strange shape, because of evolution. That's only because the structures did bend around the thalamus. And they came, depending if it was the development of reptiles, depending if it was the development of mammals. So that's why it has these strange structures. And that's why you have the fibers going through this separation, this division of the putamen, of the striatum. And you have nidus of cells here in between, particularly in the posterior part. In the anterior part, which is going to be the anterior limb of the internal capsule, sometimes some people don't even have a very well-defined anterior limb. You have much more striatum cells here than you have posterior. That's why the internal capsule. Evolution did this thing this way. Sometimes some people don't even have a very We study a lot of you have much more striatum cells here than you have, which is something very different. Evolution did this thing. Phylogeny, people don't even have a very. We study a lot of you have much more striatum cells here than you have, which is something very different. Evolution steps are repeated. We study a lot of. Okay, before beginning the lecture itself, the dissections, I want to thank Richard. Whatever you see, beautiful steps are repeated. We study a lot of. Okay, before beginning the lecture itself, the dissections, I want to thank Richard. Whatever you see, beautiful steps are repeated. Originally, we study a lot of. Okay, before beginning the lecture itself, the dissections, I want to thank Richard. Whatever you see, beautiful steps are repeated. Originally, we study a lot of. If you say the lecture itself, the dissections, I want to thank Richard. Whole life, but your brain anatomy, you have to have it since the beginning. So thanks, Richard. Thanks, Eduardo, my son who's here, that's a neurosurgeon, did University of São Paulo Medical School and stayed with Rot on a long fellowship. And he's going to be talking to you about the central core, which was his main project in the Rotten. You are here at one of the main centers of Gray's Anatomy, and we had the opportunity to write the chapter about the sulci gyre and the telencephalon, okay? And Seattle Science Foundation put this in the YouTube, and I get impressed. Everywhere I go. I went to Bangkok, I went to Malaysia a few months ago, and they had seen all these lectures. It's unbelievable. Some of the lectures have 10,000 views, and young neurosurgeons are seeing this all around. So I thank Seattle Foundation, Science Foundation for this. And now we just came with this book. I brought one here for you to see, where I did put everything I'm going to say here including the cases I'm going to be showing tomorrow, they are inside this book. So it's mostly about what I call anatomy-oriented microneurosurgery. Okay, so let's put our glasses and start our dissections here. Eduardo, maybe you can tell me when I'm reaching about 1 hour here. So, okay. Since the thalamus is the center of the brain, let's start to build the brain from the thalamus, okay? So when you look to the brain, you have to know that you have in the center the thalamus in each side of the brain, okay? Now, I always say that the thalamus is a pretty much well-defined structure. Hypothalamus is what is under the thalamus. Hypothalamus is a region. You're going to see that you have a left part and a right part of the hypothalamus, and then you have a basal part. It's a U-shaped region. Hypothalamus is a region. Thalamus is a very well-defined structure. The optic tract is the continuation of the thalamus. It's pretty much part of the thalamus, as you know. Tumors are pretty much similar, some of them, as you know. And number one, regarding architecture, always think of the thalamus as the head of the brainstem. The brainstem should morphologically include the thalamus because it's just like a body that has two heads. The thalamus is very much incorporated in the brainstem, as you see. It's the top of the brainstem, okay? And it's very much continuous with the midbrain. Complete continuous. There's no fissure here, no delineation. You have to have this in mind. And just to see this slide, to take what we can do, the medial geniculate body and the lateral geniculate body are prominence of the thalamus. You see the midbrain, you see the peduncle here, and you see the midbrain posteriorly here, the tegmentum, and this is the lateral mesencephalic sulcus. Very important sulcus. You always have a vein running here, lateral mesencephalic sulcus. Tomorrow we're going to be talking about this. And the medial geniculate body is always at the top of the lateral mesencephalic sulcus. It's a very well-defined prominence. Now, the lateral geniculate body, that is more lateral, a little bit more anterior, sometimes is flat. It's more like a region. When we'll show the dissections of the optic radiation, you're going to be seeing that the optic radiation, the visual fibers, they don't come only from this small prominence here. They come from this region. Lateral geniculate body also is more a region. But of course, you have this prominence we call lateral. Of course, you have the optic tract under here. But this slide is to emphasize that there's no division between the thalamus and the midbrain. Thalamus is the head of the midbrain. Okay? Now, when you look back here now, you know that we are in the center of the brain. You know this is the top of the brainstem, okay? And you have the midbrain continuation inferiorly. The first structure we're going to be putting around the thalamus is the hippocampal formation. Okay, we call hippocampus, if you go, another problem with anatomy is that it uses a lot of nomenclature, and sometimes use different nomenclature, different names for the same thing. But in neuroanatomy, the real name here is Amon horn. This big prominence is called Amon horn. And this is the dentine nuclei. They are very together, like two U, one inside the others. They are very, very, very together. And both of them together should be called hippocampus. But we call this hippocampus, and that's the way it is. Hippocampus is a very old structure. Very, very old, as I said. It's so old that the white matter is superficial, not interiorly. Just like the brainstem, just like the cord. Very old structures. The white matter is at the surface. Because this is cortex. This is trilaminar cortex. And of course, the axons are the surface. It's so white that it's called alveus. Are the axons of these 3 layers of neurons that we have inside here. And the Amon horn or hippocampus has a head, has a body, and has a tail. It did slip down here, you remember, during evolution, okay? And what is the fornix? The fornix is this continuation of the axons that get together. Initially, you have a very thin fornix, which is called fimbria. Then you have the crura, which is the dorsal cortex, dorsal fornix. Dorso and crura are almost the same thing. You know, crura is more like the crura, like the inferior limb. So this is the crura of the fornix. And then it gets it's flat, as you're going to be seeing, more flat. And then it gets cylinder, which is the body of the fornix. Then you have the pillar and the columns of the fornix that will end at the mammillary bodies. And of course, you have the amygdala here, anteriorly, covering anterior the head of the caudate, the head of the hippocampus, and covering a little bit also. Now, hippocampus came first. Then you did develop a gyrus along the hippocampus. Of course, this gyrus is called para-hippocampal gyrus. So this hippocampus now is part of the para-hippocampal gyrus. But the right thing to say would be the para-hippocampal gyrus is part of the hippocampus. Okay? But this is to show the superior surface of the para-hippocampal gyrus that is very flat. And it's called subiculum. If you go and look for what the words mean, you understand more anatomy. Subiculum means bed. This is the bed of the dentate gyri and also the bed of the thalamus. The thalamus is resting here. At the end of the lecture, we're going to be seeing that this is the most lateral aspect of what we call the transverse fissure of the brain. You have the PCA coming here. Okay? But at this time, just remember that the superior surface of para-hippocampal gyrus is the subiculum, and the thalamus is here. Of course, that's how nature did it. This is anterior, this is posterior, this is medial. You see the subiculum here? Very flat. This is the lingual gyrus. We're going to be seeing that the para-hippocampal gyrus is a continuation mostly of the lingual gyrus and of the isthmus of the cingulum as well. But lingual gyrus is very much continuous with the para-hippocampal gyrus. Okay? So this is subiculum. This is fimbria. This is the head of hippocampus. To see here, I did remove the amygdala. So this is anterior, this is medial, this is subiculum. You see, you can have 3 layers here. You see that the axons are here in the alveus, very white, and this is facing the ventricle inside of it. This is all para-hippocampal gyrus. Does anybody know the gyrus that is just next to it? What gyrus is this? Fusiform. What's the name of the sulcus in between? Very old sulcus, very deep. You see, almost reaches the ventricle. That's why the floor of the atrium and of the posterior horn is going to be called collateral trigon, because it's just superior to the collateral sulcus. Things have a reason to have a name. If I lift the fimbria, I'm going to see better the dentate gyri. This is the fimbrio-dentate sulcus, and this is the hippocampal sulcus that separates the dentate from the rest of the para-hippocampal gyrus. When epilepsy surgeon say we have to reach the hippocampal sulcus, they want to reach here. They want to remove the whole hippocampus, namely the head of hippocampus, part of the body, and also the dentate nucleus, because the dentate belongs to the hippocampus. Why this is hippocampal sulcus is very, very, it's not real sulcus. This is the space in between these two structures. But why is it important? Because it's the first sulcus phylogenetically. It separates archicortex from paleocortex. Okay? It separates what we nowadays call mostly allocortex from isocortex. We can talk about this later if you want to understand this better. PCA sometimes gets inside the fimbrodentate sulcus here, gets inside. This is the lateral wing of the ambient cistern. Ambient cistern is inside the transverse fissure. A cistern is a cistern. A fissure is a subarachnoid space, has a arachnoid. When I say a fissure, I'm just talking about the space between structures. The ambient cistern is inside the most lateral aspect of the transverse fissure of the brain. And along the ambient fissure, you have the PCA running. Okay? So you have to look at this and imagine all this. Of course, these structures get together along the midline. So when they are together, they constitute the body of the fornix, the body of the fornix. Now, as I said, you have to have this X-ray vision and see where you are. We're going to be seeing many times later that the fusiform gyrus is the floor of the inferior horn and of the atrium. We're going to be seeing that anatomically. the inferior aspect of the lateral ventricle is deep to the fusiform gyrus, okay? Now, you have here the parahypocampal gyrus, and this most anterior part is called the uncus. You have the apex of the uncus here. Uncus always have this triangular shape. The anterior wall is facing the carotid cistern, always. And the posterior wall here is facing the peduncle. Now, when you look to the uncus from now on, you're going to divide the uncus in two parts. The anterior part of the uncus always harbor the amygdala. And the posterior half of the uncus always harbor the head of the hypocampus. You have to have this in mind, because if you're going to operate a cavernoma in the amygdala, you can go transsylvian through the sylvian fissure. Now, if you're going to operate a cavernoma in the head of the hippocampus, you have to get inside the ventricle some way. So when you see the uncus, you have to have this X-ray vision to see these true structures there. So at this point, we're not going to see everything here. This is the uncus, okay? Anterior part harbor the amygdala. Posterior part is harboring the head of the head of hippocampus, that it's underneath the superior aspect of the amygdala. We're going to be seeing later that the amygdala is continuous with the globus pallidus. But that's just to have this initially in mind. We'll come back many times to this beautiful dissection. So the amygdala would be here. This is what you see here. Very dark, of course, is putamen. Okay, you see that the head of the body of the hippocampus is already underneath the putamen. We'll come back to this. This is the globus pallidus. So this is where the amygdala is going to be continuous superiorly. The amygdala was removed here to expose the hippocampus. So the amygdala is continuous here with the globus pallidus. This is putamen, this is globus pallidus. If you come back here, you see amygdala continuous with the globus pallidus. Okay. Relationships, architectural relationships that you have to have in mind. Now, if the so-called hippocampal formation, namely the fornix, goes around the thalamus, it's because you have a fissure in between the thalamus and the fornix. You have a fissure. What's the name of this fissure? You have choroid plexus attached here, so this is the choroid fissure. You have to understand the choroid fissure because it's going to be the main natural space to understand how to approach deep tumors inside the ventricular cavity, third ventricle, and so forth. Okay, so what is this anterior? This is the head of hippocampus. What is this here? This is the amygdala. It has to be, okay? It's anterior to that. So this is amygdala. You already know that the amygdala is continuous superiorly with the basal forebrain, let's put it this way. The lateral aspect of the basal forebrain is continuous here. Okay, so why I'm saying this at this point? Because you have no fissure here. Choroidal fissure starts indeed just here because amygdala will be covering here. Amygdala will be covering here and all anterior. And all this amygdala here is going to be continuous superiorly with the globus pallidus. So the fissure between the fornix and the thalamus has to start posterior here. It starts pretty much at the end of the head of the caudate. That's where you have the beginning, this beginning of the choroid fissure. Now, choroid fissure comes here. You see how the crude of the fornix is very flat. You see that the body of the fornix is very cylindric. And then what happens to the body of the fornix? It gets inside the brain. When it gets inside the brain as the column, pillar of the fornix, it closes the choroidal fissure. It closes the choroidal fissure. But what happens before closing? The fornix sort of detach from the thalamus before getting inside the neural tissue. So when it gets away from the thalamus a little bit, it does enlarge the fissure. When it does enlarge the fissure, what is this? This is the intraventricular foramen Monroe. Of course, you're going to have the third ventricle here, in between this thalamus and the other thalamus here. Third ventricle in between, as you know. So you see that the intraventricular foramen is very much anterior in relation to the third ventricle, okay? So this point where the choroidal fissure starts, we call inferior choroidal point. That's where the anterior anterior choroidal artery is coming in, and the inferior ventricular vein is coming out. Inferior choroidal point. So the choroid fissure goes from the inferior choroidal point until the foramen Monro. This is the choroid fissure. Of course, this is a lateral vision. Let's see a medial vision. Again, you see the thalamus, the head of the brainstem, completely continues with the brainstem, okay? And what is this here? This is the aqueduct, right? And you're going to be seeing that you have a fissure, a very deep fissure, in between the foramen Monro and the aqueduct, the opening of the aqueduct. This very deep fissure is called hypothalamic sulcus. This, it's not a fissure, I'm sorry. It's a depression here. So it's a sulcus, it's a sulcus, hypothalamic sulcus, okay? Everything anterior and inferior to the hypothalamic sulcus is hypothalamus. Everything superior is thalamus. But this is to show that crudo of the fornix comes here, becomes the body of the fornix, and then gets inside the neural tissue and go towards the mammillary body. And this enlargement of the fissure here is the foramen Monro. So if someone, when you get in surgery, you see foramen Monro, what is anterior to the foramen Monro? It's the column of the fornix. But you're going to be seeing that the column of fornix is completely continuous with the rostrum of callosum. No difference. But you have to know that the fibers closest to the ventricle, to the foramen, are the pillar of the fornix. Now, what is the back wall, the posterior wall of foramen Monro? Is this anterior part of the thalamus. And here you have 2 or 3 prominences that are the anterior thalamic tubercles. And they harbor the anterior thalamic nuclei. The anterior thalamic nuclei are pretty much only here inside the anterior thalamic tubercle. So if for some reason tomorrow they find out, discover something that you have to remove the anterior thalamic nuclei for some reason, you have just to go in the posterior wall of your foramen Monro and put your bipolar there. What you're going to be destroying there, you're going to be destroying all the anterior thalamic nuclei. That's where they are. That's where they are. Fornix and anterior thalamic tubercle. And just to see what more we have in this slide, you know that this is the lateral wall of the third ventricle. You see that the lateral wall of the third ventricle is pretty much thalamus, but it's a little bit hypothalamus also. It's the lateral part of this U-shaped region I told you. Now, you know this is the anterior wall. We're going to see better slides. What's the anterior wall of the third ventricle? Goes from the anterior commissure, they're not seen very well here, to the chiasm. This is the lamina terminalis, anterior wall. We'll come back to this, okay? So what is the floor of the third ventricle? It's hypothalamus. It's the base of the U that I told you about, okay? So anatomy is definitions. We call hypothalamus everything that is anterior to mammillary body. Everything that is posterior we call midbrain. And you see that a lot of the floor of the third ventricle is still midbrain. This is third ventricle. This is third ventricle. This is suprapineal recess. We'll come back to this, okay? So this is third ventricle. So the floor of the third ventricle is all this here. But we always think the floor of the third ventricle is hypothalamus because we know we've seen MRIs and so forth. But this is just half a floor. The other half is midbrain. So here you have the mammillary body, and then you have tuber cinereum, and then we'll have the pituitary stalk here. Okay? If you want to do a ventriculostomy, you have to be anterior to the mammillary body and posterior to the tuber cinereum and to the median eminence around the infundibular recess. We're going to be showing this later, because here you have axons that are going towards the pituitary gland, and here you don't have anything. Okay, that's why you can do your third ventriculostomy. So you have to know anatomy to figure where you are. So this is the fornix around the thalamus. This is one of Eduardo's dissection. And as I said, you see here the head of hypocampus. What is anterior? What is here? What is here? If I cut here, what am I cutting? Pardon? What is anterior to the head of hypocampus? Amygdala, you have to say it, okay? And I forgot to tell you that you have a small recess in between the amygdala and the head of hypocampus. You always have this. You see that the amygdala, again, is incorporated to the lateral aspect of the basal forebrain. You see it's incorporated. So choroidal fissure starts posterior to the amygdala. This is the inferior choroidal point area here. It's a little bit more open here. And of course, this is choroidal fissure. I forgot to say that the posterior part of the thalamus is very prominent, is the pulvinar. And you see here the lateral geniculate body here. This is lateral geniculate body. The medial geniculate body will be there at the top of the lateral mesencephalic sulcus. This is optic tract. So you see, this is the roof. This is the roof of this lateral aspect of the transverse fissure of the brain, where the ambient cistern is inside this space. So I already told you that you had as a floor the subiculum, and you have as the roof the pulvinar, and of course the lateral geniculate body and the optic tract itself. And of course the anterior choroidal artery will be coming in here. You have to have in mind this architecture. So now, when you come back here, you see the thalamus, everything we told you about the thalamus, and we know that Fornix is going around here. And you know that's a scrura. This will be the body of the Fornix going towards the foramen Monroe. And this is choroid fissure. Okay, and that's of course where the choroid plexus is. So a choroid plexus gets inside the foramen Monroe to go from the floor of the lateral ventricle to the roof of the third ventricle, as you know. Okay? So you don't have choroid plexus in the anterior horn at all. Second structure we're going to be putting around the thalamus is the caudate. The caudate also has a body, has a head, has a body, and has a tail. This is Richard's dissection. You see here the head of the caudate, the body, and the tail. While the hypocampus is going to be part of the floor of the inferior horn, the tail of the caudate is in the roof of the inferior horn. And guess what? It's going to end here sort of merging with the amygdala. He says this is the head of the caudate. Of course, this is the amygdala. This is the amygdala. Going up to the globus pallidus that was removed here. This is a piece of putamen here because it's dark. Globus pallidus will be here, okay? And you see that this tail, it doesn't really merge the fibers itself, but it's pretty much, it reaches the superior aspect of the amygdala. So you see, this part of the roof of the most anterior part of the inferior horn, you can see the head of the caudate, the amygdala, and the globus pallidus. They sort of are all together at this part of the roof of the inferior horn. Tomorrow, when we have great endoscopes that we still unfortunately don't have, we're going to be able to get there, and you see that space. You're going to see the recess in between the head of the hippocampus and the amygdala, this recess. And when you look up, you know that at this spot there, you have the tail of the caudate reaching the amygdala that is going towards the globus pallidus. Just like when you are in your room, you don't have light. If you see touch something, you know where is the other thing. That's how you should have this spatial awareness of all this that we're talking about. Pulvinar, while the Fornix, Corrado of the Fornix go medially, the pulvinar, of course the tail of the caudate goes laterally to the pulvinar. Choroid plexus, choroid fissure, foramen Monroe. We're going to be seeing that this is the rostrum of callosum later. Head of the caudate is the lateral wall of the anterior horn. Body of the caudate, it's the lateral wall of the body of the ventricle. Okay, in this preparation, we did remove the corpus callosum anatomically, okay? So you see the lateral ventricle here. You see the head of the caudate. You see the foramen Monroe here. You see the fornix going mostly posterior to the anterior commissure. We'll come back to this, okay? But I want to show two things in this dissection. Number one, you're seeing here a bundle of fibers that are running just in between the thalamus and the caudate. This is the stria terminalis. What is the stria terminalis? The stria terminalis, this is the anterior part of the uncus. Okay, you see here the apex, you see here the semiannular sulcus, where you have the anterocoroidal artery coming here. Okay, so what is inside this anterior part of the uncus again? What do I have here? The amygdala. The amygdala was shown in the early 90s that has by Johnston, that has a dorsal extension. Why this is a extension? Because you have cells in between these fibers here, other than the fibers. So the stria terminalis is a dorsal extension of the amygdala and ends at a small nucleus here at the base of the head of the caudate that is called the bed nucleus of the stria terminalis. Okay? So this is a dorsal extension. And then an Argentinian that worked at UVA with Dr. Leonard Heimer. Some of you might have met him. Okay, Jose de Olmos was an Argentinian, and he described a ventral extension of the amygdala that comes to the temporal stem, something that we're going to be seeing later, to the ventrostriatum area, and also ends at the bed nucleus of the stria terminalis. That's why they came with this concept, Leonard and Olmos, of extended amygdala. That the amygdala, because you have, it's not only fibers, you have cells in between the fibers. So it's a dorsal and a ventral extension of the centromedial amygdala, only the biggest part of the amygdala, centromedial amygdala, okay? So you have this, the amygdala would be a whole system around the thalamus and hypothalamus, and they call this the extended amygdala. But at this point, I want to show the stria terminalis, because we neurosurgeon, we read the things in neuroanatomy, or the neurologist talking about this, we have to know where it is. We have to know where it is. So stria terminalis, of course, we have to know what is the stria terminalis, but we have to know where it is. So I said that the stria terminalis is running in between the caudate, you see caudate veins here, and the thalamus that is here. What else is running here? What else is running in between the caudate and the thalamus? No, in the surface. Which vein? Thalamus striate vein. So you see that the thalamus striate vein that you see in surgery go towards foramen Monroe, goes around and goes to the floor, to the roof of the third ventricle, becoming the internal cerebral vein, okay? And when it goes around the foramen Monroe, it receives the septal vein from anterior. You remember this. So you see the thalamus striate vein when you get into the ventricle. The stria terminalis is running underneath, along inferiorly to the thalamus striate vein. So next time you see the thalamus striate vein, you know that the stria terminalis is running underneath. It has any function? I don't know. As far as I know, it doesn't have any function. It's a vestigial structure that is there, but it's there. And ends at the bed nucleus of the stria terminalis. In this slide, I'm showing also the fornix. This is the crura, this is the body. And of course, it goes mostly posterior to the anterior commissure, and then projects and ends in the mammillary body. Mammillary body, it's easy to dissect a tract that goes from the mammillary body until the anterior-thalamic nuclei. Anterior-thalamic nuclei, posterior wall of the foramen Monroe. This is anterior wall of foramen Monroe, this is posterior wall. Okay, from the anterior-thalamic nuclei, everything that gets to the thalamus is projected to the cortex. So, anterior-thalamic nucleus project to the basal frontal brain and to the cingulum. Cingulate gyrus has inside the cingulum, goes around the cingulum. Cingulum is continuous inside the parahypocampal gyrus, okay? And we'll end here at the amygdala. The amygdala has a dorsal extension, goes around the stria terminalis, and puts all this thing together. So now this part of the circuit that goes along the fornix and then goes to this region and then come back as the cingulum. What system is this? That projects here again. It projects here to the cingulum, goes to the cingulum, okay, and then goes to the parahypocampus and comes back as the fornix, and again to the mammillary body, anterior tubercle, cingulum. What circuit is this? The Papez circuit, okay? 1936, he described this circuit. He thought it was the circuit of emotions. His original paper is called Circuit of Emotions. but then we know nowadays that it's used particularly with the short-term memory. Okay, you see that these structures are all together because they arise together along evolution. That's how they came. Okay, so you can see here the puppet circuit. Now, we did cut here with a knife, a sushi knife, to show that the corpus callosum is the roof of this ventricular cavity we're talking about. And as the ventricular cavity is always covered by the ependyma, that's why the light is reflecting here, because you have a membrane, which is the ependyma, very thin membrane, okay? Now, we always think about the corpus callosum along the midline. Everything that is facing down is called rostral. So this is rostrum of callosum, okay? This is genu, or knee of the callosum. This is body of the callosum, and this is the splenium. Now, the corpus callosum opens to all to both sides, and what I came to tell the grad students that the corpus callosum is like a butterfly. This would be the body of the butterfly, and this would be the wings of the butterfly, the fibers that are opening up and down. So we have to figure the butterfly going with the wings up and down to understand the three-dimensional anatomy of the corpus callosum. Very complex, because opens everywhere. And the corpus callosum has 200 million fibers. 200 million fibers that are arising from are putting together both hemispheres. So it's very well defined in the midline, but laterally it opens as a bunch of fibers going up and down. So you can see here, corpus callosum would be covering here. This is the caudate. This is choroid plexus. If I cut here, gentlemen, what I'm cutting if I cut here? Laser point is over the thalamus. And what is over the thalamus at this point? What do you have here? Covering the thalamus posteriorly. Everything that is lateral to the choroid fissure. You have the crudo of the fornix. When you look at here, you have to know that the crudo of the fornix is here, covering the thalamus. Now, laterally to the choroid plexus, this is thalamus. And this part of thalamus is called lamina fixa in anatomy, neuroanatomy, because this is the point where the diencephalon is attached to the telencephalon. So that's why this area here is called lamina fixa. This is thalamus. If you cut here, you're already cutting thalamus. If you go more anterior thalamic nuclei, the posterior part of the foramen Monroe. Now, if you go everything that is medial is fornix. Here, the body of the fornix, and here, all crudo of the fornix. It's flat, always covering the thalamus. Now, you see that the corpus callosum is covering the body of the ventricle and the anterior horn. Now, when it comes to the atrium, it's covering the atrium here, the posterior part of the ventricle, but it's going inferiorly, it's going underneath. So always, if someone asks, what are the walls of the atrium? And of course, of the posterior horn, because the posterior horn is an extension of the atrium, are the fibers of the corpus callosum. They go superior and inferiorly. They go all around the atrium and the posterior horn. You have to know this in order to understand and to know, because it's difficult to dissect, to see this. Okay? So here is just superior, but here goes inferior also. And then it's going to end covering part of the temporal horn, the posterior part of the temporal horn, called the tapetum. We're going to show the tapetum later. It's our posterior callosal fibers. We talked about corpus callosum, rostrum, genu, body, and splenium. We talked about the fornix, thalamus. I forgot to talk about the massa intermedia. Some people do, some people don't have, some people do have. Functionally, it's not important, just white matter fibers. You can see here the hypocampus sulcus that will be coming towards the opening of the aqueduct. That is not shown here. But at this point, I want to emphasize the relationships between the corpus callosum and the fornix. You see that the body of the fornix, it's not attached to the corpus callosum, and of course, neither the pillar or the fornix. This is the anterior wall of foramen Monroe. Look, see foramen Monroe here? This is anterior wall. Posterior wall, anterior thalamic nuclei. Now, you see that the crude of the fornix is completely attached to the inferior surface of the callosum. Completely attached. So, we're going to be seeing this from here, posterior here. So, this is a splenium. So, you see these two things that are attached to the inferior surface of the corpus, the splenium, are the crude of the fornix. Crude of the fornix. We're going to be seeing from here now. Crude of the fornix. In between the two crude of the fornix, we have fibers that go from one crude to the other. Every fibers that go from one side to the other are commissural fibers. So what's the name of these fibers here? Fornicial commissure or hypocampal fornicial, because they are putting together functionally both hypocampi. So if you cut the splenium, other than be cutting the very important splenial fibers that you know about, you're going to be cutting also the hypocampal commissure, which put together both sides. Midbrain, aqueduct, substantia nigra, peduncle, lateral mesencephalic sulcus, medial geniculate body. I'm not seeing very well, but I know that it's there. I know this is the medial geniculate body region because it's at the top of lateral mesencephalic sulcus. So what is this? Pardon? Ulvinar. So what space is this? You have again, you see the map? It's a very old map, but there's something that looks like a boot. So that's Italy, because you know, okay? So I'm showing a space, a natural space, in between what? What structure is this? Again. This is the crude of the fornix, and this is the pulvinar of the thalamus. So what space in between the fornix and the thalamus? The choroidal fissure, of course. So this is the posterior part of the choroidal fissure. Tomorrow we're going to be showing a tumor that is both in the pineal region and in the atrium. Of course, the tumor opened the choroidal fissure. So in order to understand the anatomy of the tumor, you have to know anatomy. Tumors like to go through natural spaces. Of course, it's easier for them. Okay? So this is the choroid fissure seen from behind. So the crura is attached to the inferior part of the splenium, and then you have a membrane that is attached to the corpus callosum and to the body of the fornix. What membrane is this? You know that everything that is anterior to the anterior horn, to the foramen motor, is anterior horn. We're talking about midline here. So we're talking about the medial wall of both anterior horns. What is this? The septum pellucidum, of course. Septum pellucidum. We are building everything here. So, septum pellucidum is here. So septum pellucidum ends where the fornix meets the splenium, right here. From here until you have septum pellucidum. So septum pellucidum is the medial wall of the body of the ventricle and of the anterior horn. Let me emphasize that everything that is anterior to the thalamus is anterior horn. Everything that is superior to the thalamus is body of the ventricle. Everything that is posterior to the thalamus is atrium. And that the occipital horn or posterior horn is an extension, posterior extension of the atrium. Inferior horn will be down there. So we are now seeing that anatomy, a lot of definitions. By definition, the atrium starts at this point where you have no more septum pellucidum. So this is atrium. If you have a tumor here, already atrium. If you have a tumor here, body of the ventricle. So we're seeing that the septum pellucidum is medial wall of the atrium and medial wall of the anterior horn. What is the anterior wall? What is the anterior wall of the anterior horn? No, not rostrum. Anterior wall. Genu. And what is the floor of the anterior horn? What is the floor of anterior horn? Rostrum. Rostrum is over there. Tomorrow we're going to see this in surgery. Rostrum. What is the lateral wall of your horn? You have to know this by heart. Head of the caudate. This is all Dr. Rotton's description, which is very good in the sense because he puts, again, lateral wall of the atrium is body of the caudate. The floor of the atrium is both the thalamus, lamina fixa here, and the body of the fornix more medially. If we come to the atrium, what is the anterior wall of the atrium? Is the pulvinar of the thalamus, and is the crude of the fornix that is going around here. Everything that is covering the thalamus medially to the choroid plexus is fornix. So you have to know this is fornix. This is not this is fornix covering the pulvinar. The real thalamus is here and underneath the choroid plexus. Now the medial wall of the atrium, you have this. This is the insertion, the posterior part of the hypocampus, of the tail of hypocampus, that reaches the medial part here of the wall of the atrium. And then you have a big bundle of fibers that is part of the splenium that is bulging here. This is the callosal bulb of callosum. You know that the posterior part of the corpus callosum, namely the splenium, harbors the forceps major, while the genu of the callosum harbors the forceps minor. So the hypocampus and the bulb of callosum are the medial aspect, the medial wall of the atrium. And a little bit more posterior, you have in this depth here, believe me at this point, that this is the depth of the calcan fissure. And you have neurons here. What's the name of this bulging here in the ventricle? Calcar avis, exactly, calcar avis. Just related also with the callosal bulb. So here, you see better what I'm talking about. Medial wall, you have the hippocampus, this is hippocampus, coming here to the medial wall of the atrium. Posterior, you have the bulb of callosum, and more posterior, you have the calcar avis. You always have this angle here, always, in between the hippocampus and the bulb of callosum. And this is the upper aspect of the floor of the atrium, which is continuous posterior with the occipital horn. See how flat this is? And it's always a triangle here. This is why this is called collateral trigon. Collateral trigon, because it's triangular. And as you already saw, the collateral sulcus is running underneath here. The more deep the structures are, the more constant they are anatomically, of course, because they are older. Okay? So you always have this image here, this collateral trigon. Now, you understood the posterior wall of the atrium, you understood the medial wall of the atrium, you understood the floor of the atrium and of the occipital horn. Let's see now the floor of the inferior horn. Let's take this in another specimen, and let's see what's the floor of the inferior horn. You see, now I've seen the inferior horn here. Well, if I come back here, I see a flat surface, which is the collateral trigon, which is the floor of the atrium and the floor of the posterior horn. If I go more anteriorly, namely inside the inferior horn or temporal horn, I see that this collateral trigon becomes bulging here, just like the hippocampus medially. So it changed its name to collateral eminence, because this is an eminence. So the floor of the atrium is collateral trigon. The lateral aspect of the floor of the inferior horn is collateral eminence. And what is the medial part of the floor of the inferior horn? Hypocampus. Don't be afraid, please. You know, I have to feel that you are really understanding what I'm saying. So the medial part of the floor of the inferior horn is the hypocampus, or amon horn if you want. And this is collateral eminence. The floor of the inferior horn, this is very important because you're going to be there in many tumor surgery. You have to know what is the floor of the inferior horn, okay? Now, we saw all these structures related with the ventricle. Now, we have something in between here. This is all called the central core. Eduardo is going to be giving a lecture of this, and very important to understand the central core of the brain, of course. But at this point, what is this here? I had called it, this is the thalamus, and this is the putamen. Putamen together with globus pallidus is called lentiform. Globus pallidus and putamen has nothing to do one with the other. And I mean functionally, anatomically, evolutionary, whatever, embryologically, but they are together. So together, they look like a lens. They are called together lentiform nucleus, okay? Now, you see here the lentiform nucleus, and you see that it has an internal capsule and an external capsule. So always understand the internal capsule as the internal capsule of the lentiform, mostly of the putamen. An external capsule of the putamen of the lentiform nucleus. Always understand this, okay? Now, the internal capsule has 5 parts: an anterior limb, a genu, a posterior limb, a retrolentiform, and a sublentiform. The external capsule is more anterolateral here and has claustrofocal fibers. We're going to be talking about this later. Eduardo is going to repeat by the time. Anatomy is repetition. That's why we're going to be repeating here these 2 days. Very much the important structures for you to have it by heart then. Okay. Internal capsule. What do you have in the internal capsule? Architectonically, again, see that the genu is very much related with the foramen mauro. Very next to the foramen mauro. Very next. I'm going to be showing later today that you have in the cortex a few beautiful U's. When you see a beautiful U along the midline interhemispheric fissure, this is the paracentral lobule. And I'm also pointing very certainly because paracentral lobule is always sitting over the atrium. It's very posterior, very related with the atrium at its depth. So this is precentral gyrus, this is postcentral gyrus. What is that internal capsule? Internal capsule, you have fibers that are coming from the thalamus, okay? Thalamic cortical fibers, cortico-thalamic fibers, they go back and forth, so they go everywhere here. Frontal region, central region, parietal, occipital, temporal, you have cortico-thalamic fibers, or thalamo-cortical fibers, better. Now, all internal capsule also has fibers that come from the cortex and go to the cerebellum, to the pons. Cortico-pontocerebellar fibers. All the way here from all the different regions. Now, the anterior limb doesn't have very important functional fibers. Then when you come to the genu and to the posterior limb, okay, you have motor fibers, precentral gyrus, motor strip. What part of homunculus is here? Pain-filled homunculus. Down here where you have what part of the homunculus? The face and the oropharynx, right? So these fibers, they are going through the genu. And then this is superior frontal sulcus. Superior frontal sulcus, precentral sulcus always interrupted, and you have a region here. What's the name of this region? Omega region. What do you have in the omega region here? Hand. So hand, upper limb, goes to the anterior half of the posterior limb. Paracentral lobule, what type of fibers you have here? Going to the Exactly, they are more posterior. Okay, now, the superior thalamic radiation that goes towards the postcentral gyrus, it's very important, is in the posterior part of the posterior limb. So these parts are very important in internal capsule. Retrolentiform, very much related with auditory fibers that are going towards the Heschel gyrus. We're going to be seeing very much the Heschel gyrus. And more inferior, the sublentiform or sublenticular part, very much related with the optic radiation, just go anterior as the Meyer's loop, covering the ventricle here and go towards the calculation. Of course, you have to understand very well the internal capsule. But at this point, I want to emphasize that the internal capsule is the internal capsule of the lentiform nucleus. That's how you should see it. And the geniole, very adjacent, very much related with the foramen Monroe. This is most center structure. This is the thalamus. So the fibers that are going from the thalamus to the cortex are the more medial fibers of the internal capsule, of course, because the thalamus is more medial. So its fibers are medially positioned. Okay? Anterior thalamic radiation, superior thalamic radiation, posterior thalamic radiation. Okay? This is only names. But this is all thalamocortical fibers. If you remove this dissection, was done by Dr. Leonard Heimer in Charlottesville. If you remove the thalamus and the fibers, you already start seeing the lentiform nucleus. This is all putamen here. It has the shape of the putamen. Okay? And you see that the fibers that are coming from the cortex and going towards the peduncle, this is peduncle. They go posteriorly, of course, like a fan shape, because they are going to constitute the peduncle. Now, this is the upper limit of the lentiform nucleus. This is the inferior limit. So only these fibers here, anatomically, they are named internal capsule fibers. When they come from here superior, in the books, they are the same fibers, but in the books they are called corona radiata or centrosemiovalle, okay? Because they are superior to the superior limit of the lentiform nucleus. And the fibers, when are inferior, they are peduncular fibers. These are the peduncular fibers. Of course, are the same fibers, different names, depending on their topography. But you should understand this architecture of the fibers. Almost finishing this first part. So we're talking about this lateral part, which is central core. Eduardo is going to talk with us about details. He's going to repeat for you to understand. We're talking about this part here. Because we already talked about the inner structures related with the ventricles. But what is this here, please, quick? What is this? White, medial part of the inferior horn. What is this? Has something anterior to it. What is this? Hypocampus. Please don't be afraid. And what is this? Amygdala. So you look at it, you have to name it. So you know that the caudate is going from here. What is this? I would say that this is at least, it's where the tail of the caudate should be here, coming towards the amygdala. Okay. Inferior horn, the atrium will be here. This is putamen. This is putamen. So these fibers, projection fibers, remember Minor, Theodore Minor, he was psychiatrist, neurologist, neuroanatomy. He studied very much, and he named the fibers. He was the one that divided projection fibers and association fibers. If it's association between the two hemispheres, commissural fibers. If it's association in the same hemisphere, intra-hemispheric association fibers. Okay? So these are projection fibers because they are going up and down. You have thalamocortical, and you have the internal capsule with the rest of the internal capsule fibers, because the thalamocortical fibers belong to the internal capsule, as you know. Okay, so superior to the limital lentiform, they are corona radiata, centrosemiovalle. If you remove the lentiform, then you're going to be seeing the internal capsule of the lentiform which is inside here. We're going to see later. Now, since the internal capsule has this fan shape because it's going towards the peduncle, if you see anteriorly, they are not separated. Beautiful dissection from Richard. You see that the internal capsule did split the striatum. This is the whole striatum was here. It did split evolutionary. Everything that is medial is the caudate. Everything that is lateral is the putamen. And this is the ventral striatum. Very important for neuropsychiatry, for everything, for insular tumors getting this region. We're going to be talking about this. This is the ventral striatum or nucleo accumbens, not splitted by the internal capsule. You see anterior commissure here. You see fornix. You see olfactory tract. We're going to be returning to this. So if you remove the putamen carefully, you see globus pallidus inside. And this is internal capsule. The same fibers here, different name, but the same fiber. And these fibers are going towards the peduncle, are going to be peduncular fibers. And of course, anterior to the globus pallidus, very constant, always the old anterior commissure. Anterior commissure. Of course, this is amygdala. Of course, this is hypocampus. Globus pallidus. Amygdala is going towards the globus pallidus, as you already showed you in another section. I did put again here the putamen because I'm going to be coming laterally. We saw the internal capsule. Again, internal capsule. Now, putamen. And what is superior, lateral to the putamen? This is the external capsule. Most of the fibers of the external capsule are claustrofugal fibers. But anteriorly, you have a very well, let's say, enlargement of these fibers, bigger fibers. And the more anterior ones are the uncinate fascicle, and the more posterior one is going to be the IFOF, inferior frontal occipital fascicle. Very important. We're going to be showing this. Eduardo is going to emphasize this. So this is all external capsule. If we come more, we're going to be seeing the claustrum together with the subcortical white matter of the insula, which is, and superior to this, you have, superior to this, you have the cortex of the insula. So what is the, just underneath the cortex of the insula, how you call this? Extreme capsule, right? But you already, it's together a little bit of the claustrum, because the claustrum is so thin that it's difficult to dissect laterally when you go sagittally, okay? Now, what is the extreme capsule? The extreme capsule is nothing more than the subcortical white matter of the cortex of the insula. You have U fibers in between the gyri, every part in the brain, also in the insula you have. So those U fibers, the subcortical fibers of the insula are called extreme capsule, right? So these are all the structures we did put together. And you see that they constitute altogether a very well-defined block that Dr. Rotten called the central core of the brain. You see that its anterior half is related with the head of the caudate. Its posterior half is related with the body of the caudate and with the thalamus. And the foramen Monroe is a little bit anterior to the half of the insula. So when you see the central core, you know that you have the internal capsule, the external capsule, of course, the putamen. And you see here the inner aspect of the choroidal fissure. This is Eduardo's dissection, to emphasize that everything is in between, just to complete this first part. Let me just show this last part here, that it's very interesting, and we save some time. This is the lateral wall of the third ventricle. Lateral wall of the third ventricle. So this is thalamus. This is the aqueduct. Can you see here this depression here? Hypothalamic sulcus. So everything that is inferior is hypothalamus. Okay? So this is the lateral wall of the third ventricle, massa intermedia. This is the roof of the third ventricle. Superior have the body of the fornix. Crura is here, attached to the splenium. This is all body of the fornix. This is pillar of the fornix. Anterior wall of foramen Monroe. Posterior wall hidden here, anterior thalamic nuclei. This is stria medullari thalami. Stria medullari thalami. It comes from the back of foramen Monroe until the abenular area, which are small bulgings here, prominence that are called the abenular, where you have the abenular nuclei. Abenular nuclei. Stria medullaris. You have a space. This is all roof of the third ventricle, all roof. But you see that you have a space in between the stria medullari thalami and the fornix. This is inside the roof of the third ventricle. This is a space that is continuous posteriorly with the quadrigeminal cistern or pineal region, namely the pineal cistern and quadrigeminal cistern. And this space gets inside here. Does anybody know the name of this space? This is a cistern here. That it's an anterior expansion of the quadrigeminal cistern. What's the name of the cistern? Velum interpositum. So the velum interpositum cistern is inside the roof of the third ventricle. And what do you have in here? And what else? Exactly. These structures are all inside the floor of the roof of the third ventricle. This is lateral wall. You see here the mammillary body. You see here the pituitary stalk going towards the pituitary. So this is floor. Now the floor, this is the anterior half of the floor of the third ventricle. You already know that you have a posterior half, that it's inside here the ventricle. Inside here the midbrain. This is peduncle on one side. You cannot see peduncle very much. What is in between the both peduncles? What's the name of it? Interpeduncular fossa. Interpeduncular fossa. I'm emphasizing this to show that the roof of the interpeduncular fossa is the posterior half of the floor of the third ventricle. You have to know this. It's difficult to see. Surgery, you're never going to be seeing this. But you have to know that the posterior half of the floor of the third ventricle is the roof of the interpeduncular fossa. Architecture, relationships, right? If you see what is inside the wall, of course, this is infundibular recess. This will be supraoptic recess, okay? This is the pineal. We're not seeing very well the recesses here, but I understand the pineal. The pineal cistern, the pineal gland, is posterior to the third ventricle. It's attached to two commissures. Superiorly, it's attached to the commissure of abenula, going from one side to the other. Inferiorly, it's grabbing, holding the posterior commissure. Posterior commissure, let me emphasize, I always forget to say something. This is the aqueduct, opening of the aqueduct. The inferior border of the opening of the aqueduct is midbrain itself. The superior aspect of the aqueduct is the posterior commissure. Posterior commissure is right here. So when you see the aqueduct, everything that is superior is posterior commissure. Everything that is inferior is midbrain itself. Now, and then here in this side you have the abenula. And in our side you have other abenula of the other thalamus. In between the abenulas you have the commissure of the abenulas. Now the pineal gland is attached to the brain, holding superiorly the posterior commissure. The commissure of abenula, and inferior the posterior commissure. And the space that is in between both of them is the pineal recess. Of course, because I'm the pineal, this is the arm that is holding the posterior commissure, this is the arm that is holding the abenular commissure. So this space is the pineal recess, okay? And superior, you have the space over the abenula. So this is pineal recess, this is infundibular recess, this is supraoptic recess. This is the what? Hippocampus sulcus. Okay. So now let's dissect here to see what we have inside as well. And we'll finish this presentation. And you have this beautiful dissection here done by Richard. You see the pineal gland. Oh, here you see this is the posterior commissure here. This is the superior part of the aqueduct, superior limit, superior border of the aqueduct. Okay? And the abenula will be coming from here, where the stria medullaris thalamus ends at the abenula. And there will be a superior arm holding the abenula commissure and an inferior arm holding the posterior commissure, as you can see here. The space in between is the pineal recess. Of course, this is the thalamus. This is the stria medullari thalami. This is all space that belongs to the roof, namely to the velum interpostum cistern. That is continued with the pineal cistern here, posterior. We're going to be talking about this transverse fissure of the brain again, which is important to understand the architecture of all this thing. Thalamus. Fornix ending at the mammillary body, coming mostly posterior to the anterior commissure. Mammillothalamic tract of Vic d'Azur, I didn't emphasize the name of this tract, that is going to the anterior thalamic nuclei and is going to project in the cingulum. And here you have the subthalamic nuclei. Subthalamic nucleus is just underneath the thalamus. I never had seen this dissected as Richard did. And this is the interpeduncular nucleus. So this is the red nucleus. I'm sorry, the red nucleus, nucleo hubro. And this is subthalamic nucleus. And in between, in the middle of the midbrain, you have the interpeduncular nucleus, nucleus interpeduncularis. And you have a tract that comes, you don't have to know this, but just let's start. You have a tract that comes from the abenula to the interpeduncular nucleus, in this tract. It's called fasciculus retroflexus fasciculus of who? Of minor again. So this is heterocephalus fasciculus of minor that comes from the abenular region toward the interpeduncular nuclei. You have here the red nucleus and you have here the subthalamic nucleus. This is all inside the wall, the lateral wall of the third ventricle. Posterior wall, you have the epithalamus, which is the pineal part of the third ventricle, the pineal area of the third ventricle, posterior to the third ventricle. Of course, this is the third nerve, you see. This is the third nerve that will be arising within the interpeduncular fossa. Infundibular recess, the infundibulum going towards the pituitary. Supraoptic recess would be here. Of course, this is chiasm. This is optic. This is chiasm, optic chiasm. Subthalamic nucleus, retroflexus fasciculus of minor. Posterior commissure, which is the superior border of the opening of the aqueduct. So if you come here for the third ventricle, for example, you have figure where I am here, okay, mammillary body. You see that anterior to the mammillary body, you have this very thin floor. That's where you do your third ventriculostomy. More anteriorly, you have the tubercinarium area, where you have axons from the hypothalamus that are going around the infundibular recess, which is called the medium eminence. And these are all axons that are going along the pituitary stalk, the infundibulum, to the pituitary gland. Anteriorly, you would have the supraoptic chiasm, the supraoptic recess. Now, here you can see beautifully the opening of the aqueduct. I told you that the posterior limit of the opening is the midbrain itself. And what is this here? Posterior commissure. The posterior commissure is the superior limit of the aqueduct. So this is all midbrain. This is the roof of the interpeduncular fossa. You have to have this architecture. This is the roof of the interpeduncular fossa. This is the roof of the interpeduncular fossa. Now, this area here that is more thin is the roof of the interpeduncular cistern, right? Where you do your opening and you're going until the prepontine space to make sure the CSF is running from the third ventricle inferiorly. And that's why you find the basilar tip here. And that's why basilar tip aneurysms very easily bleed inside the third ventricle. Oh, here you see beautifully. Aqueduct, midbrain. What is all this here? Pardon? And what is this here? A benular commissure. So what's the name of this space here? In this one? A benular recess, suprapineal recess, which is the benular recess, suprapineal recess. This is the pineal recess. This is roof of the interpeduncular fossa. You don't want to get in here. The red nuclei would be here. And just to end, this is a beautiful, yesterday I was talking about this good friend of mine, Aldo Stam, is an ENT guy that operates school base with us. And he did remove this huge craniopharyngioma, post-chiasmatic craniopharyngioma, transphenoidally endonasal. He did a beautiful job, and you can see all this anatomy here distorted. I just wanted to finish with this beautiful removal. Oh, and this is mass intermedia. It's the same case we're talking about. You see both foraminal Monroe here. You see the posterior commissure here. Opening the aqueduct would be here. So now let me really end with this concept of the fissures of the brain. This is not important at all, but it's important to think, to have in mind the architecture of this, because we have mostly to understand the brain in relation to the natural spaces, to the sulci, to the fissure, to the ventricles, because that's where we want to get. At least it are the landmarks related to the places we want to go to, okay? So Professor Yassergill, who is indeed the father of this, all these anatomical concepts that gave rise to microneurosurgery, okay? He comes with these concepts that we have three main fissures. One fissure very easy to understand, interhemispheric fissure. Other fissure, very easy to understand, Sylvian fissure. You know all these very well. Now, you have this concept of the transverse fissure that was described by an old French neuroanatomist called Bichat. That is the space that are medial to the temporal lobe, lateral to both peduncles, go posterior to the brainstem, and gets inside the roof of the third ventricle. So, the most lateral aspect comes from the inner aspect of the Sylvian fissure, then comes having medially the peduncle, superior the optic tract that I showed you, and inferior the subiculum, where the PCA is coming. That's where the ambient cistern is. Now, both ambient cisterns come along both sides of the transverse fissures, okay, and they meet posterior to the pineal, where you have the pineal region, where you have the pineal cistern. And as I showed, the pineal cistern continues anteriorly along the roof of the third ventricle as the velum interpositum cistern, where you have all the things that you named. So this space is the transverse fissure of the brain. So if you understand the choroidal fissure and the transverse fissure of the brain, you understand all the spaces that tumors can go through, extrinsic tumors, epidermoids, for example, they love to go through this space. You can understand how you can get there, opening the choroidal fissure, for example. I'm going to be showing later if we have time. You get into the third ventricle. If you open the choroidal fissure very posterior, you get into the pineal cistern. If you open the choroidal fissure in the body of the ventricle, you get in the velum interpositum cistern. Okay, so these relationships between the choroidal fissure and the transverse fissure will show all the spaces you can go through, and hopefully one day we'll be really able to navigate with an endoscope to this space. And technology will make it possible, but anatomy will lead you there to know where you are.